All right, part 2 of chapter 6, section 2. Really just looking at the two main topics of biofilms and then what's called quorum sensing. So biofilms, right? Bio is life, film is a covering when we think about it in terms of biology. And those are places for bacteria to live and to amplify and just expand at a high rate due to the protection that this biofilm gives it. All right, so you've got the life part of the biofilm, and then you have the protection part, that film. And as the biofilm is starting, you're going to have what's called a pioneer colonizer. All right, that pioneer colonizer is a particular type of bacteria, and it doesn't really matter which one it is. It's just going to be the first one. And it attaches to a surface, so it does have to have a means of attachment. So maybe it has fimbriae that will allow it to lock onto its surface. And then other microbes are going to attach to those bacteria, those pioneer colonizers, or a particular substance, whether it's a sugar or a protein substance that those pioneer colonizers have laid down. All right, and so then there is just this process that occurs. Figure 6.8 gives you the steps in the formation of a biofilm. And we talked about the pioneer bacteria that colonize a surface. And so up there at step 1, you have these little pink rods. All right, so let's say these guys are gram-negative, just because they're pink. All right, they're going to be the first inhabitants of that biofilm. And they will come and sit down on a surface. All right, now that surface can be your shower curtain, the surface of your sink. It can be your teeth. It can be a valve, say a heart valve, or a joint, or a artificial joint that has been inserted surgically. All right, and so what happens is those pioneer bacteria are going to secrete extracellular material, and that is the globby blue stuff that you see here. And then in addition to that protection matrix material, they're also going to start secreting chemicals, and those are the little red dots. All right, now those little red dots are the chemicals that are being produced in terms of those pioneer bacteria wanting to communicate with each other or other species. All right, so in step 3, it says in many but not all biofilms, other species join and then may contribute to the extracellular matrix, right, and or participate in quorum sensing. So here you have the pink rods, and now you have purple rods. You have the red dots, which are the red communication signals, and then now you have blue dots. Those are going to be other communication signals. And it's in this biofilm, this basically bacterial community, where you start to see what's called quorum sensing. And the quorum sensing is just that. The bacteria sense that there is a group of bacteria there. All right, so it senses there's a quorum, and that term has always kind of confused me exactly what is a quorum. It's a particular number of a group that's supposed to be present, and if you have a quorum, if you have a certain number of the members of that group, then you can officially have that meeting or whatever the case may be. But quorum sensing for bacteria is that the bacteria will start to act different when they get into an actual community like this biofilm. So their genes are regulated differently. That means they can express different proteins. Maybe it's these red chemicals or these blue chemicals. And it's just like a very tight-knit community, where some organisms do one job, some organisms do another job, and they get the signal through this process of quorum sensing that is the job that they're supposed to do. All right, so a couple of things about the biofilm. All right, with this matrix material, this blobby-like material, this is protection. All right, you can kind of think about it as an umbrella, and when you're in the rain and you pull out an umbrella, you take out the umbrella to stay dry. The rain hits the umbrella and it rolls off, and hopefully it stays away from you. The bacteria are going to be protected by or from antibiotics. They're going to be protected from the immune system, because the immune system cells are not going to be able to get through that matrix and start to phagocytize and destroy the bacteria. The antibiotics are not going to be able to penetrate that biofilm. And so you can think about it as an umbrella. When the antibiotics are the rain, well, the bacteria pull out their umbrella and the antibiotics just simply roll right off. And it never gets to the bacteria, and so therefore it's hard to treat the biofilm. And then in some cases, the bacteria, what you see here, can actually leave the biofilm and start to be the normal bacteria that you saw up here, where they just simply replicate like normal. And so then they can get into the blood, and that person can have a systemic infection, or they can just simply go to a different area of the body and possibly start a new biofilm, but not necessarily. All right, so a person can go from not knowing there's a problem or just feeling very fatigued if, say, they had a biofilm on a heart valve, or pain in a joint if it was on a joint, to feeling very ill because of that systemic infection and bacteria in the blood. Another issue that can arise with biofilms is that part of this biofilm itself can actually break off, so you have a chunk of it that breaks off and starts moving through the blood. And then when it comes to a smaller vein or vessel, capillary, whatever the case may be, it can get stuck, and it can basically occlude the blood from moving anywhere else. So that would be a major issue for like the deep veins in your legs. It can be an issue if that chunk of biofilm gets into the lungs and causes a blockage within your vasculature of the lungs. And then obviously within the brain, it can block the blood vessel or the blood flow there and end up allowing a person to experience a stroke. So how do physicians treat biofilms? It can be a tough battle, right? Especially, well, if it's on a heart valve, sometimes that heart valve will be completely destroyed, and the patient has to have surgery to have it removed and a replacement valve put in. That's not always the first solution or treatment option. The person will probably be put on IV antibiotics, and these drugs are going to be at high concentration in the blood, directly into the blood. They can be for months at a time to try to clear that infection out. I know of an individual whose brother had to have hip replacement surgery, and this poor guy, it was, I don't know, a year-long process, because he had surgery to have the hip replacement, and then a biofilm developed, and he was in bed on antibiotics in a lot of pain for several months while the physicians were hoping to get rid of the biofilm. And it didn't work, and he had to have a second, maybe even a third hip replacement surgery, because at some point you just can't do anything and you simply have to physically remove that biofilm. And for a synthetic joint, it's going to be removing that joint and having to go through that entire process all over again. So it's very taxing on a person, physically, obviously, emotionally, definitely, because, you know, there always is that worry that an individual can die because of the infection or have other issues like the chunks breaking off and limiting blood flow, having a stroke, having other damage occur in the body. So you want to be careful to avoid this at all costs. Now, common examples of biofilms are going to be the plaque and the tartar on your teeth, and sometimes the dentist or the dentist hygienist will have to scrape plaque off your teeth. No, tartar. Tartar is the hard junk on your teeth, and that is plaque that has hardened. And what's going on there, that is a biofilm. And the bacteria are living underneath it, and they are going through metabolism. And one of those metabolic byproducts is going to be acid, and so the bacteria secrete acid to the surface of your teeth. That degrades the enamel, and then you have cavities. So the way to get rid of that is to scrape your teeth. And hopefully if you have good dental hygiene, you won't have that issue develop. But even the best of us can sometimes have issues. Alright, so here is actual definition of quorum sensing. Alright, used by bacteria to interact with members of the same species, as well as members of other species that are close by. And this is just simply by their chemical communication. And it's interesting looking at these features here, the structure of the biofilm. Alright, it's a large complex community. The bottom may have a very different pH and oxygen conditions from the surface. So here, if you think about this being the bottom of the biofilm, the organisms can actually do very different things than the organisms that are up here. Even though you have purple and pink guys up top, you have purple and pink guys at the bottom. The actual environment can be very different. And if the environment is different, then the bacteria are going to do different things, and that is the idea of quorum sensing. Right, and it is a partnership, right, among multiple microbial inhabitants. Yes, you got the pink and purple guys on the previous slide, but there could be red, blue, black, white, orange, all of those different organisms, because there could be, huge numbers of different organisms that are part of one biofilm. And it could just be a couple. So it doesn't have to be multiple, but it certainly can be. And as I mentioned, it cannot be eradicated by traditional methods. You have to do major medical treatment to get rid of these biofilms. Alright, so concept check. Which of the following describes an association between microbes in which one organism is benefited and one is harmed in some way? So your options are mutualism, synergism, commensalism, parasitism, and antagonism. And, alright, one organism is benefited while one is harmed, and the answer is going to be parasitism. And that is the end of Part 2, Section 6.2.